So in point mutation, we discuss about three types. That is silent mutation, no effects, that is harmless. Missense mutation, example is sickle cell anemia. Then finally nonsense mutation, that is example is thalassemia. Defect is it acts as a terminator codon and finally stop the protein synthesis. Second type of mutation is classification, frame shift mutation. The name itself, frame of the nucleotide sequence is changed. So the definition is when there is insertion or deletion, this word is very important. When there is either insertion or deletion of one or two nucleotides, okay, in DNA, automatically produces altered mRNAs, right? So that is why the meaning is altered reading frame of the mRNA. Altered reading frame of, that is the frame will get defective. Altered reading frame of the mRNA. Actually, we have two types of frame shift mutation. One is either insertion, another one is deletion. Okay. So that is the effect is completely irrelevant protein with altered amino acid sequence is produced. Okay, this is also another example for thalassemia. Second one is sometimes prematurely terminated protein also synthesized in this frame shift mutation. First one is completely irrelevant protein with altered amino acid sequence. Second one is prematurely terminated protein also synthesized. Okay, so the insertion, the example is thalassemia again, whereas the deletion, the example is cystic fibrosis, one of the important disease involved in the child age group, that is cystic fibrosis. That is example for the deletion type of frame shift mutation, whereas insertion type of frame shift mutation example is thalassemia disease, that is one chain defective, either alpha chain or beta chain defect. Is there any test to find out these mutations, right? Yes, we have one test to find out the mutations, that is it is a simple test to identify the mutations is Ames test, that is somebody says Ames test, some book says Ames test. AME, spelling is AME, that is Ames test, which is a test for the identification of mutations. So frame shift mutation and point